And I would like to first start by level setting of what metabolism is, and to help understand what metabolomics and why we think metabolomics is the key to understanding biology, health, and disease. Metabolism is the conversion of food and fuel to energy to run all cellular processes. And these enzyme-catalyzed reactions allow organisms to grow and reproduce to essentially maintain the biology of life. And it's the intermediary and chemical endpoints of these enzyme-catalyzed reactions that are called metabolites, or biochemicals, that I will use interchangeably. Metabolomics is the large-scale study of these metabolites within all biosamples, which include cells, biofluids, tissues, and organisms. And metabolomics represents the quantitative assessment of these changes in these biochemicals to understand how they influence biological systems. And metabolomics is really the keystone of systems biology, in that it integrates both genetic and non-genetic influences to provide a definitive representation of a phenotype. And on the top part of the slide, shown in purple, are what is typically thought of the flow of genetic information, or the central dogma of molecular biology, that DNA goes to RNA goes to proteins. And what I like to point out is that metabolomics integrate this information from these upstream modalities to integrate both this information as well as the diet, lifestyle, microbiome information on the bottom part of the slide shown in blue. But on the top part of the slide, what I like to say is that there's a level of complexity with these omics modalities that confounds our ability to understand how they operate across different model systems, for example, from yeast to mice to humans, and even within individuals, such that genetics, for example, they display a wide range of genetic variations. Transcripts, RNA, go through a wide array of post-transcriptional modifications, such as splicing and processing, and proteins undergo a bewildering array of modifications post-translationally, myristylation, phosphorylation, nitrosylation, etc., that confound our ability to understand how these operate in different model systems. Unlike metabolites that are conserved from bacteria to humans, and that structural conservation implies functional conservation, such that the TCA cycle, for example, is conserved in bacteria as it is in humans, and that helps us understand how they work across different model systems. But also, it's the integration of xenobiotics, exposure-related metabolites and biochemicals, microbiome, and lifestyle that makes metabolism and metabolomics a really unique modality to understand how these are all integrated to provide a comprehensive view of a phenotype. Microbiome, for example, is a buzzword these days, in that bacteria and bacterial-produced metabolites influence a wide variety, almost every variety, of human health and wellness and transition to disease. And we're able to capture this information, as I said, to provide a comprehensive view of a phenotype. But that phenotype has a research abstract, in that what we really want to understand is real-world outcomes. And metabolomics is the key to driving optimum insight from all these upstream omic modalities, as well as external factors such as exposure, microbiome, and lifestyle, to provide real-world outcomes for what's going on in an individual, such that we can functionally map upstream omics modalities such as genetics to understand what's going on in genes and genetic variants and how they influence health and wellness and real-world clinical outcomes, and all these other non-genetic influences that also affect, like the microbiome, that affect health and wellness in a real-world status. And now I'd like to provide an example of why we feel metabolomics provides really unparalleled insights that are unseen with genomics. And this is an example of sickle cell anemia, which is still a highly prevalent disease today. For example, in Africa alone, in Nigeria, 150,000 children are affected with this on a yearly basis, and over 15,000 individuals, patients, die worldwide as a result of this disease. And this is despite knowing the molecular basis of this disease has been known for over 60 years. It was in 1956 that Vernon Ingram identified a mutation in the beta globin gene that changes a glutamine to a valine at position 6. And this induces conformational changes in the beta globin that induce a change in the red blood cell shape, so that it produces a sickling shape, which is shown on the bottom part of that slide. And despite knowing the mutation for over 60 years and the genetic basis of this disease remains untreatable to this very day, and the mechanistic basis of this disease are unknown. And it wasn't until a metabolomic study was done recently, flash forward 60 years, That insight was provided into the metabolic basis of this disease that provides therapeutic opportunities and insights for how to treat these individuals that are currently facing an untreatable situation. Metabolome profiling identified perturbations in a number of pathways, including adenosine, lysophospholipid or fatty acid metabolism, 2,3-diphosphoglycerate, as well as amino acid metabolism, shown here, aspartate, on the right part of the slide, that showed that four major pathways are perturbed relative to control individuals that are shown on the left part of that panel, such that this provides therapeutic opportunities for these individuals. For example, adenosine at the bottom part of that panel is a receptor-mediated system, and being receptor-mediated system, this provides therapeutic options for drug targets to go after. And in fact, this is under review today. So it's metabolomic profiling that provided unique and powerful insights to new therapeutic opportunities to treat patients that had been essentially clinically left behind for over 60 years.